In this video we will look at the Nobel Prize in Chemistry 1915 awarded to Richard Willstätter for his researches on plant pigments, especially chlorophyll. Here you see chlorophyll, as you know, it's green and it's in the leaves of plants. And you can see why it is green. It absorbs blue and red parts of the white sunlight and it deflects green. Then some work information from the Nobel Prize site. The leaves of green plants derive their color from chlorophyll, which plays a key role in enabling plants to transform water and carbon dioxide into carbohydrates and oxygen with the help of sunlight. From 1906 to 1914, Richard Willstätter researched the chemical composition of chlorophyll. He discovered that the chlorophyll of different plants or species is the same, but is a mixture of two different types which he produced in pure form. He showed that magnesium is an essential part of chlorophyll structure and pointed out chlorophyll's relationship with the hemoglobin in red blood cells. This is coming from the official site, you have the link. Then some biographical information summarized by me, also coming from the official site. As a young man, he, as Willstätter of course, studied principally the structure and synthesis of plants alkaloids such as atropine and cocaine. In this, as in his later work on quinone and quinone-type compounds, which are the basis of many dyestuffs, he sought to acquire skill in chemical methods in order to prepare himself for the extensive and more difficult work of investigating plant and animal pigments. He enjoyed his work in Zurich so much that he did not think of those years as a waiting period until he was called back to Germany in 1912. In the 2 short years before the outbreak of the First World War, he was able with a team of collaborators to round off his investigations into chlorophyll and in connection with that to complete some work on hemoglobin and in rapid succession to carry out his studies of anthocyanins, the coloring matter of flowers and fruits. These investigations into plant pigments, especially the work on chlorophyll, were honored by the award of the Nobel Prize for Chemistry in 1950. The investigations into photosynthesis and into the nature and activity of the enzymes were precursory of modern biochemistry. At that time, the method so far developed of concentrating enzymes through adaptation did not make it possible to attain to the crystallized enzymes. In this connection, Willstätter carried out important studies of adsorbents, metal hydroxides, hydrogels, and silicic acid. In addition, he was quick to give his attention also to the problems of theoretical chemistry. Thus he achieved the first synthesis of cyclooctatetraene and went on to compare it with benzene. So also he set up experiments to produce cyclobutadiene. Willstätter's career came to a tragic end when, as a gesture against increasing anticism, he announced his retirement in 1924. In 1938 he fled from the Gestapo with the help of his pupil A. Stoll and managed to immigrate to Switzerland. This is a summary, of course. If you want to read the complete biographical information, follow the link to the official website. Here you can see that there are two chlorophylls, A and B, in plant, and you see they absorb different wavelengths. But as you can see, not here, no green. Then we continue with a summary of the award ceremony speech given by Professor O. Hammerstein, and the summary is made by me. By its property of making possible the assimilation of carbon dioxide under the influence of sunlight, and hence introducing the synthesis of organic substance in the green parts of plants, chlorophyll. The elucidation of the nature and the mode of operation of this substance is therefore a task which is of the highest degree of importance. The difficulties in the field have been so great that until very recently they have prevented a successful study of the problem of chlorophyll. Willstätter is the first, jointly with several of his students, to have been successful in overcoming these difficulties. By the new and important discoveries resulting from these investigations, he has been able to elucidate in all its essential parts the question of the chemical nature of chlorophyll. It is true that earlier investigators had observed that chlorophyll contains magnesium, besides other mineral substances. Willstätter was the first to recognize and prove with the complete evidence the fact that magnesium is not an impurity, but is an essential part of the native pure chlorophyll. He has shown that magnesium is held within the chlorophyll molecule in a manner which is very similar to the way in which iron is held in hemoglobin. This bond is so firm that magnesium is not liberated even by the action of a strong alkali. On the other hand, it can be removed by an acid without injury to the remainder of the chlorophyll molecule. Willstätter has made use of this circumstance to test to what extent chlorophyll can be the same in different kinds of plants. He showed that the chlorophyll was the same in all the kinds so far examined. It is a mixture of two somewhat different but yet closely related chlorophylls, one of them being blue-green, the other yellow-green, and the former occurring more richly in the leaves than the latter. The fact that chlorophyll in the ordinary sense is a mixture of two green pigments had, it is true, already been shown to be probable by Stokes in 1864, and both Zet and Maslewski had brought forth important support for this view. It is Willstätter, however, who had here produced the certain and conclusive proof. To prepare chlorophyll in an unchanged pure state and in such quantities that it can be the subject of complete chemical analysis has of course been one of the most important tasks of the chlorophyll research. At the same time, it was one of the most difficult of all. Willstätter has also been able to prepare the two above-mentioned different types of chlorophyll in a pure state and so supply exact proof of their existence. In doing so, he had been able to carry out a thorough investigation of the large amount of the various derivatives which can be produced from these two different chlorophylls, and as a result of this means he has brought a desirable clarity to the field of chlorophyll chemistry, which was previously very complicated and confused. He has also created new and rich possibilities for further fruitful research in this field. The most important part is, nevertheless, that relating to the detection of the chemical structure of chlorophyll. He has shown that chlorophyll is an ester, which on saponification with alkali can be split up into a previously unknown alcohol called phytol, which represents about 1/3 of the molecule, and a color component called chlorophyllin, containing magnesium. He has more closely investigated these two components, both individually and for their transformation and composition products. The splitting up of chlorophyll can also take place as a result of action of an enzyme occurring in the leaves, which he called chlorophyllase, and hence he has been able to elucidate the nature of the crystallized chlorophyll. He has established that it's not the pure unchanged natural pigment in the leaves. The crystallized chlorophyll is a laboratory project, an alkyl ester which lacks phytol. The amorphous chlorophyll containing phytol is the unchanged natural pigment in the green parts of the plant. A very important section of Willstätter's work on the chemical nature of chlorophyll is represented by his investigations into the color components, the chlorophyllin and other phylins, and derivatives from it. These investigations are also of particular interest with regard to the question of the relationship between blood pigment and chlorophyll. From the iron-containing red blood pigment, hemoglobin, substances can be prepared purple in color and free from iron, which are known as porphins. And the one which has been known the longest is this, hematoporphyrin. A substance is very closely related to this, phyloporphyrin. Schunk and Maslewski have shown that a chemical relationship does exist between blood pigment and chlorophyll. It is Willstätter who has conducted the complete conclusive investigations. In these investigations, which concern the pigment nucleus both in chlorophyll and hemoglobin, he has made several new and important observations regarding the pyrroles and the composition in the nucleus. In particular, however, he has shown that from these two pigments the same parent porphin, a thioporphin, can be prepared, whose molecule has retained the essential characteristics of the pigment nucleus. By doing this, he has produced the most interesting and decisive proof of the relationship between the most biologically important pigments in nature, hemoglobin and chlorophyll. He has also prepared in a pure state and studied exhaustively yellow pigments, the so-called carotenoids, which occur together with chlorophyll in the leaves of plants. By means of the results obtained by regarding both these yellow pigments and the chlorophylls, he has paved the way for new biological researches into the part played by the different leaf pigments in assimilation of carbonic acid. He has also studied with great success another group of plant pigments, namely the blue and red pigments of flowers, the so-called anthocyanins. As a result, the anthocyanins have been shown to be glycosides which can be split up into a kind of sugar, in most cases glucose, and a color compound, an cyanidin. Willstätter has elucidated the chemical structure of these cyanidins. He has proved in what their difference consists in the various flowers of fruits, and has also proved their close relationship with the yellow pigments occurring in natures of the flavone and flavonol group. He has shown the dependence of the flower pigments upon the reaction of the plant sap, and has thus explained how one and the same anthocyanin can have a different color in different flowers. By extending to the yellow pigments of flowers as well, and by quantitative determination of the anthocyanins, he has shown that the difference in the color which the flowers assume depends on several different circumstances. Well, this was a summary of the award ceremony speech given to Willstätter. As I said, it's a summary. If you want to read the complete text, then you have to follow this link. Here you see, by the way, a chlorophyll molecule. You can read that the black balls are carbons, so white is red, hydrogen, red is oxygen, turquoise is nitrogen, and orange is magnesium. Then you can see how it's built up. And here you can see what kind of pigments are in different blood pigments, leaf pigments. Here you can see chlorophyll A and chlorophyll B. We already saw them, both greenish, slightly different. As you can see, there are more. Next week, we will go into the Nobel Prize lecture given by Richard Martin Willstätter. For now, thank you for watching, and maybe until another time.